Hello everyone, welcome to our weekly reading session. Everybody is welcome to join. The details are in the description below. In today's talk, we are hosting Professor Peter Ku, and he is presenting his groundbreaking research titled Evaluating the Representational Power of Pre-trained DNA language models for Regulatory Genomics. In this paper, the researchers are trying to establish the capabilities of DNA language models, and we are evaluating whether these pre-trained DNA language models can genuinely understand the complex rules of genomic regulation, or they are just language models that are just scratching the surface. So, in this talk, we are going to find out and get ready to find out surprising findings uh, and find whether these models are good in interpreting biological sequences. Let's dive in.
Today, I'll be presenting new work from my lab on exploring the representational power of pre-trained DNA language models for regulatory genomics. This work was led by a former grad student in my lab, Amber, who is now at InstaDeep. So simply put, large language models have been transformative for NLP, and their real-world impact is just starting to be realized. They have also started to make an impact in the sciences, especially in protein biology. These protein language models are trained in a self-supervised manner on a large corpus of protein sequences gathered from public protein and metagenomics databases. And just from the pre-training alone, the language models seem to learn a lot of information about proteins, such as statistical relationships like conservation, coevolution, and phylogenetics. And the utility of these so-called foundation models is that their representations, which encode many properties of proteins, can be utilized for a wide variety of downstream prediction tasks, including predicting protein structures, predicting the effects of variants or mutations, predicting protein function, as well as for protein design. And this is especially important within intrinsically disordered regions where current structure-based models like AlphaFold struggle. But while proteins are the workhorses of the cell, they only comprise 1 to 2% of the genome. And DNA is the master blueprint for an organism. And as it goes with the central dogma, DNA encodes RNA, and then mRNA translates into proteins, which carry out most functions in a living organism. Importantly, DNA also encodes for gene regulation, controlling when, where, and how much of a gene is expressed. This regulation allows cells to respond to spatial temporal cues from the environment, differentiate into specialized cell types, and orchestrate complex biological processes. Now, to model the genome, one approach is to train large language models, treating each nucleotide as words, similar to how protein language models treat amino acids as words. The two main approaches for language modeling of biological sequences are masked language modeling and causal language modeling. In masked language modeling, a random set of input tokens are masked, and the model is trained to predict the masked positions based on the surrounding context. This ensures that the model learns bidirectional statistical dependencies between positions in a given sequence. In causal language modeling, the model is trained to predict the next token based on the previous tokens. So this encourages a unidirectional contextual understanding. However, one benefit of this approach is the ease of sequence generation. So again, language models provide a powerful way to learn contextual embeddings of nucleotides. The embeddings consider how the nucleotides interact with the context of the sequence. So by training on genomes, a language model should be able to identify at a high level different regions of the genome, such as regulatory elements and non-coding or encoding regions. And in principle, it should also be able to capture meaningful patterns within regulatory elements, such as sequence motifs, as well as their complex dependencies with other regulatory elements. From the coding regions, it should also be able to learn the codon language of proteins and the conservation and coevolution of amino acids they encode for. And genomic language models have exploded in the past few years. Here's an incomplete list, and you can see that each gLM, or genome language model, differs from one another in the various modeling choices, such as in tokenization strategies, language modeling objective, base architecture, and training data. For instance, in terms of tokenization, you can choose single nucleotides, overlapping or non-overlapping k-mers, or bipair encoding, which are variable size k-mers. For the base architectures, transformers are typically preferred, but there are also convolution-based models and state-space models, or some hybrids among these. For training data, gLMs are trained on either just a single genome or multiple genomes within a species, or genomes across diverse species. From these genomes, they can consider the whole genome or be specialized to specific regions like just UTRs or just the coding regions. However, despite the excitement of these genome language models, it's not really clear what they are capable of because they have yet to be thoroughly evaluated. Oftentimes, they demonstrate slightly better predictive performance against weak baselines on arbitrary genomic prediction tasks. And these benchmarks are what the ML community is currently overfitting to. But even in these uninspiring predictive tasks, the genome language models are fine-tuned on each downstream task. This is very different from protein language models, whose representations from pre-training are already encoding for a vast amount of useful information. And so one can achieve quite a lot with zero-shot or few-shot learning settings. And so this leads us to the central question of this study: how much regulatory biology is learned in a non-coding genome by these genome language models during their pre-training? Now, to give you some more biological motivation, regulatory elements in the non-coding genome, like enhancers and silencers, play a crucial role in controlling gene expression. Understanding these elements is key to deciphering many biological processes, from development to disease. For transcription to occur, specific proteins must bind to the DNA and make the region accessible, creating cis-regulatory elements such as enhancers and promoters. But it's not just about one cis-regulatory element. Multiple cis-regulatory elements interact over long distances to control which gene is expressed. This ensures that the right genes are turned on and off at the right time, enabling cells to differentiate, respond to signals, or maintain normal function. So what we expect the gLMs to be learning during pre-training on entire genomes is to capture a wide repertoire of regulatory sequence patterns, such as transcription factor binding motifs. Now, the lexicon of motifs should span the entire corpus of relevant motifs across the human genome for any cell type. This, in essence, would mean that the gLM has learned a foundational understanding of regulatory motifs that are broadly useful for various cell types and conditions. Then, during fine-tuning on a specific functional genomics dataset, the representations can be used by a downstream model to upweight and downweight the motifs that are relevant. And fine-tuning would then allow the model to select the task-specific features instead of learning everything from scratch. And this would make the model more efficient, reduce the amount of training data needed, and improve overall performance. However, this is only a hypothesis. We don't actually know how much regulatory knowledge gLMs are actually learning during their pre-training. Are they learning the biological features that matter, or are they simply providing a better initialization for fine-tuning? This leads us to our central question: how much foundational regulatory information is being learned during the pre-training? To answer that question, we designed the following experiments. For each of the genomic language models, which I'll mention in the next slide, we probed the embeddings from the penultimate layer. These embeddings represent the features learned during pre-training. And importantly, the model weights were not fine-tuned. So these embeddings purely reflect what the model has learned during pre-training. And we then use these embeddings as inputs to downstream models to evaluate their performance on functional genomic prediction tasks. And as a baseline, we compare the performance of the gLM embeddings to one-hot encoded representations of the DNA sequences. And our benchmark spans 6 different downstream tasks, but today I will focus on the results from the first 3 tasks. Here's an overview of the gLMs we explored in this study. The models we focused on include the nucleotide transformer, GPN, high-NID DNA, and DNA-BERT2. These represent a wide diversity of modeling approaches, including the base architectures, language modeling objective, and tokenization strategies. And so, for instance, nucleotide transformer was trained on non-overlapping 6-mers, and it used a transformer encoder, a BERT model, and trained with mass language modeling. And it was trained on hundreds of different species. And GPN is a custom model that we trained on a small number of genomes for model organisms. And the tokenization strategy is nucleotides. And the structure is a convolutional neural network, and it's also trained with mass language modeling. High-NID DNA is a state-space model that uses the high-NID operator. You can think of this as an implicit convolution. And it also uses the nucleotide tokenization. And in this case, it's trained with causal language modeling objective. And DNA-BERT2 uses bypair encoding, and it also uses a BERT-style transformer encoder and mass language modeling, and is trained on hundreds of different species as well. Now, since all of these models were exposed to the entire human genome, they should be applicable to prediction tasks focused on the non-coding regions of the human genome. And so the first task involves a quantitative relationship between cis-regulatory sequences and their ability to drive transcription using the lenti-MPRA assay. And I'm not going to go over the specifics of this assay, but at a high level, lenti-MPRA is a technique to measure the activity of cis-regulatory sequences. Unlike traditional MPRA, which is an episomal reporter assay, lenti-MPRA integrates variable sequences into the genome through lentiviral transduction, providing a more stable and realistic environment for observing regulatory function. Here, the cis-regulatory sequences are 230 nucleotides long, and are associated with a single scalar target that represents the transcriptional activity of that sequence. And we explore 2 lenti-MPRA datasets for different cell types, HepG2 and K562. And for each of these, there are over 100,000 training sequences. First, we explored different probing approaches, including probing with linear models, as well as nonlinear models that consider the entire embedding. For both CLS and mean embeddings, the CLS token, these represent a summary of the gLM embeddings into a single vector. We applied a ridge regression and an MLP model. These treat the embeddings as fixed representations without any positional information. However, the full embeddings retain positional information across the sequence, and so we applied a convolutional neural network. And this CNN is a basic model with just a few layers. It's a typical model that we commonly come across in regulatory genomics. And as you can see from the results, using a CNN trained on the full gLM embeddings yielded the best predictive performance across all models. And therefore, for the remainder of our analyses, we're just going to stick with the CNNs as the main downstream model. Next, we compared how models using gLM embeddings performed against those trained with one-hot encoded sequences using the baseline CNN model. And as a control, we considered representations from a randomly initialized DNA BERT2 model, and that's denoted as random here. We also included SAY. This is a multitask supervised model trained on over 20,000 chromatin profiling datasets across more than 1,300 cell lines and tissues. For consistency, we treated SAY like the gLM embeddings, extracting embeddings from the penultimate layer and feeding them into the same downstream CNN. And all models, whether they're based on embeddings or one-hot sequences, shared the same CNN architecture. And for comparison, we also included a more advanced one-hot model, Resonant, which incorporates residual connections for better feature extraction. And we also included MPRANN, which was from the original study. And this is also for comparison. And as you can see from these results, the gLM embeddings in blue did not provide any real substantial performance improvement over the one-hot trained CNN, CNN in orange. And however, the supervised SAY embeddings in green significantly improved the performance. So these results suggest that gLM embeddings may not capture informative motif patterns in regulatory sequences, and they might even be losing some critical information compared to the naive one-hot encoding. Now, to explore this further, we moved on to the next task, predicting transcription factor binding using ChIP-seq data. And here's an overview. ChIP-seq data measures protein DNA interactions and can be processed into a binary classification task, where the input sequences come from accessible regions, and a 1 label is given to the sequences if it overlaps with the ChIP-seq peak, and a 0 label is given if there was no ChIP-seq peak associated with that accessible site. And this enables learning about the sequence elements that are predictive of TF binding. We ran this analysis independently for 10 different transcription factors in GM12878 cells, data taken from ENCODE. And the long story short is we found similar trends across the board. And here I'll just be showing the results for nucleotide transformer. And as you can see, the gLM embeddings perform similar to the one-hot sequences, albeit slightly worse. And here, each dot represents a different ChIP-seq experiment. Now, however, based on these results, it's not clear whether the gLM embeddings are learning important sequence statistics, such as motifs, or if the gLM embeddings are similar, or like, well, no, if the gLM embeddings are simply not losing too much information about nucleotide identity, which the downstream CNN is then able to learn motifs from. So to investigate this further, we compared the performance of MLPs trained using the CLS token, both of which summarizes the entire sequence. And so the CNN trained on the full embeddings performed better than the MLP, indicating that positional information indeed matters here. And as a control, we also trained an MLP just using dinucleotide frequencies of the sequence, and found that the performance was quite similar to the CLS token on average. And so this suggests that the CLS token might mostly encode low-level sequence statistics, such as dinucleotide frequencies. However, it remains unclear whether the full gLM embeddings are encoding higher-order information like motifs, or if the downstream CNN is constructing those motifs from these basic sequence statistics. Now, to help us address this question, we turn to attribution analysis. And attribution methods are post hoc explainability tools to give insights into the importance or sensitivity of input features on model predictions. And in practice, this can be accomplished by perturbing the input data and monitoring how the predictions change. In genomics, a natural perturbation is a single nucleotide mutation. So by systematically mutating each nucleotide in a given sequence, we can map out an in silico mutagenesis map, and we can represent this as a sequence logo, as shown here at the top. Over the years, several alternative approaches have emerged, such as calculating the gradient of the prediction with respect to the input sequence. This is called a saliency map. And this too gives, the sensitivities of each nucleotide, and it can be represented as a sequence logo as well. And there are many other popular methods as well, including deep SHAP, smooth grad, integrated gradients, deep lift, and many, many more. And attribution methods have gotten a lot of flack over the years, especially in the computer vision community. And that's because a perturbation to a single pixel of an image of a dog is still an image of a dog. So the importance of pixels lives in a very redundant regime in images. In addition, features in an image exist at multiple scales. So pixel level explanations are not informative of whether it's texture, the shape, or other intermediate characteristics that drive the model's predictions. But in genomics, a single nucleotide perturbation can dramatically change the functional activity of the sequence. So in practice, attribution maps have been quite informative in genomics. And here's an example of an attribution map for an enhancer sequence in Drosophila S2 cells. And there are many putative binding sites that resemble known motifs from a motif database, such as GATA, AP1, and STAT. Note that the motifs here at the top are from the Jasper database. And the attribution maps are the ones shown below. And you can see that they have a striking resemblance to one another. Now, to investigate whether biological features like motifs were learned during the pre-training phase or by the downstream models, we employed saliency maps. For comparison, we use one-hot trained CNNs as a baseline, since their saliency maps reveal known motifs. Next, we plotted attribution maps for the same sequence using two different gLMs, GPN and nucleotide transformer, which is just NT here. Here, we systematically masked each input token one at a time and acquired the prediction of the masked token. We then calculated the entropy of the predictions. Then we scaled the observed token according to the entropy. And so for GPN, the tokenization is nucleotides. And for nucleotide transformer, the tokenization is non-overlapping sixmers. And that's why they look a little blocky. So it only shows the importance of tokens as a positive magnitude, but the directionality isn't considered in this entropy calculation. So that's why they're all positive here versus the saliency maps. Nevertheless, unlike the one-hot CNNs, the attribution maps from these language models did not show clear signs of identifying meaningful sequence patterns like motifs. This suggests that gLMs may not be learning well-defined motifs during their pre-training. Now, to further explore this, we repeated this analysis, this time using the downstream CNN that was trained using the gLM representations. Here, we basically multiplied the CNN gradients with the entropy term to scale the token level attributions. Now, the GPN CNN combo seems to better reflect the motifs there's some overlap with the one-hot CNN saliency maps here. However, nucleotide transformer, which deals with sixmers, is still challenging to interpret. And this indicates that it's likely that the downstream CNN is doing most of the work in learning cell type specific motif features, not the pre-trained gLMs. And so next, we moved on to zero-shot variant effect prediction. Predicting the effects of variants is a common downstream goal for foundation models in genomics. And to assess the accuracy of the variant effect predictions, we use the CAGI5 dataset, which is comprised of experimentally measured saturation mutagenesis via an MPRA for different regulatory elements. This dataset provides experimental measurements of variant effects for all possible single nucleotide variants in selected regulatory elements. So for our zero-shot strategy, we explore two methods inspired by the original gLM publications. The first approach, called log ratio method, involves passing in a sequence with a mass token, and the gLM then predicts the mass token, and we calculate the log ratio between the predicted probabilities of the reference allele and the alternative alleles. The second approach passes two sequences through the model, one with the reference allele and one with the alternative allele. The gLM generates embedding vectors for both, and we calculate the variant effect by computing the cosine distance between these two vectors. In both cases, the gLM was not trained on the MPRA data. All predicted variant effects are based solely on the knowledge the gLM acquired during pre-training. So we did this for nucleotide transformer, GPN, and hyena models. We then calculated Pearson correlation between their predicted variant scores and the experimental measurements from the CAGI5 dataset. As a comparison, we also evaluated zero-shot performance of pre-trained supervised models as well. Here we have SAY and Informer, which is another sequence to function prediction model. You can see that gLMs are underperforming in variant effect predictions, especially compared to these supervised models. And this result was expected to some extent, given that these gLMs were trained without any cell type specific information, which is crucial for accurately predicting variant effects in different cellular contexts. So next, we considered the performance of CNNs that were trained on the lenti-MPRA dataset using the gLM embeddings. Now, for these models, we still pass two sequences into the gLM, one with the reference allele and one with the alternative allele. We extract two separate embeddings for these sequences, but now we feed them to a downstream CNN model, and the CNN makes two predictions, and we compute the difference between these predictions as the variant score. As you can see, adding cell type specific information through a downstream CNN model significantly improved the variant effect prediction performance. And this suggests that the downstream training on lenti-MPRA data was necessary to capture key cis-regulatory features that are specific to each cell type. However, it's important to note that the CNNs trained with the SAY embeddings provided impressively strong performance. And even though the same CNN was used for each of these different embedding methods, the supervised SAY embeddings appear to be much more beneficial than gLM embeddings when it comes to variant effect predictions. And this highlights the value of supervised learning in encoding cell type specific regulatory information, and suggests that gLM embeddings, while useful, may not capture the same depth of knowledge without fine-tuning. So in summary, we've seen that genomic language models face significant challenges in predicting regulatory functions in the non-coding genome, which is in contrast to their success in modeling proteins. So why do these models struggle with regulatory DNA? One key reason is the inherent structure of protein sequences versus non-coding DNA. Proteins have a clear start and end, with shared grammatical structures like secondary structures and protein domains. And these are highly conserved across species, both at a sequence level and covariation levels. In contrast, non-coding DNA is far more complex and variable, with regulatory elements like enhancers and silencers being sparsely distributed among seemingly random regions of the genome. On top of that, regulatory DNA presents a significant class imbalance problem. Informative regulatory elements are rare and scattered among long stretches of uninformative sequences. When models are tasked with predicting large numbers of arbitrary nucleotides without any structure, they can only be successful through memorization rather than learning generalizable biological patterns. And finally, the relationship between DNA sequence and cellular behavior is extraordinarily complex. Even though every cell in an organism shares the same DNA, different cell types exhibit vastly different spatial temporal behaviors due to complex regulatory signals. And this makes it challenging for models trained solely on genome sequences to capture the nuances of cell type-specific regulatory mechanisms. And this is hard because they don't consider any functional information measured under different cellular contexts. Now, while it should be possible to learn all of this regulatory information across all cell types from just the DNA alone, in order to do so, I believe that there must be sufficient genetic variation to learn this stuff from. Unfortunately, humans don't have that much variation, and the inclusion of primates or other similar closely related species can certainly help. But as we go further down the evolutionary tree, the divergence might simply be too great for that data to be useful. Now, moving forward, we could address these challenges by incorporating functional genomics data into the pre-training process, or conditioning the model on cell type or tissue-specific annotations, or focusing the gLMs on specific elements throughout the genome as specialists, and then building upon them in a hierarchical manner. Now, before I finish, I want to highlight that my group has been very hopeful for large language models in genomics. But due to these challenges I outlined in the previous slide, we have become increasingly excited about the prospects of an alternative approach, namely discrete diffusion. We have a preprint out where we adapt score entropy discrete diffusion for regulatory DNA sequences, and we find it to be actually quite compelling. And we think that our DNA discrete diffusion model is able to get around the issues of existing generative models because it follows a denoising process of mutating nucleotides. And this generative process doesn't have to deal with the vast junk DNA, which is what plagues other generative models that rely on a reconstruction-based loss. Instead, it simply has to mutate nucleotides toward a desired functional activity, much like evolution. In this case, the model is trained to rapidly evolve random sequences into functional ones. And you can read more about it in our initial preprint. Now, stepping back at a high level, there's a lot of excitement for the development of virtual cells, a digital replica of living cells. And the basic approach is to train a machine learning model on vast amounts of multi-scale biological data, including genomics, epigenomics, transcriptomics, proteomics, within single cells and as part of tissues using spatial data. And the current approaches are focused on building foundation models separately for each of these different modalities, building a protein foundation model, a transcriptomics foundation model, a genomics foundation model, and so forth. And the hope is that their representations can be fused somehow to integrate the modalities into a more comprehensive understanding of core biological processes, from gene regulation, regulatory networks, and understand how the environment influences signaling pathways. And this paradigm enables performing counterfactual experiments at scale to predict how cells respond to perturbations such as genetic mutations, gene knockouts, and drug responses. And while my lab focuses on just one part, genomics, I can tell you that we are far from building foundational representations of the genome, especially in regulatory regions in the non-coding genome. And so despite the excitement of these so-called digital twins of the cell, we are far, far from this pipe dream. And with that, I'd like to thank, Amber, who is the lead person, who pioneered this work. She did a huge number of different experiments, and it was really informative because this kind of work is really important because we're finding the limitations of current genome language models, and in identifying their limitations, we can try to address their problems. Thank you.
Thank you, Professor Peter. I hope you can hear me. We have a terrible network problem here. Multiple people have been admitted and they remained in the waiting room. They couldn't join. So I don't know whether I can be heard.
Yeah, you can. Yeah, I can hear you.
Okay. Thank you very much. Prof, you really, this paper is like a bombshell with what we expect with the power of language models, that the way the research community has been pushing out language models. And my first question is, when you look at the gLM, it has been lagging behind compared to the pre-trained CNN. And yes, you've put out the possibility of the cell type data during training within the different proteins and the non-coding DNA. But inherently, the way we know CNNs, like they have been powerful in computer vision and image classification. And I don't get the idea like how they come to overpower language models in this scenario. So I could have missed this, but how come that CNNs are overpowering language models? Yes, we've seen how language models are struggling the other side, but like, why is that? Probably I missed that, but why?
Yeah. That's an excellent question, and the reason why is primarily because of the structure of our data. So even though we just have like a string of DNA sequences, right, of different nucleotides, which seems like a sequential task might make sense, or like a sentence, treating it like NLP makes sense.
Yes.
It also makes sense to think about them like images too, because in regulatory DNA sequences, it's like a 1D image where we might have binding sites for proteins, and proteins bind to specific types of DNA sequences with higher affinity. And those binding sites are in different locations in different sequences. And so learning like convolutional kernels are very good at finding these patterns that have translational equivariance. And so that's why convolutions have a very strong inductive bias to capturing these motif-like patterns in but like what I think moving forward, what we need to do to build language models in genomics is to improve the inductive bias. So not just considering transformers from scratch, but maybe we need some hybrid models like convolutional transformer models. And I know there's some early works on this, but none have been really that compelling yet.
Wow, that's interesting. Like, yeah, when you say the hybrid, like a convolution combined with a transformer, and this is something really to think about. And talking about treating the sequence as an image, at the surface, when you say it, it appears abstract for me. But if we are to design these large language models in terms of genomics, can attention improve? Because this is a sequence, multiple like codes.
Yeah.
Can't attention-based language models perform better than a plain language model, maybe?
Yeah, so attention like is certainly going to be very, very beneficial because the genome, like if we just focus on proteins, proteins are really dense and rich with information. Uh,
Yes.
like you won't just throw in lots of amino acids. Like evolution won't do that because they're like there's no benefit to it, and you in fact you might be at a disadvantage if your proteins are unnecessarily big. And in the genome, like it's more like you have coding regions and then you have non-coding regions, and protein coding regions are only 1% of the genome. Like the vast majority of the genome is, to me, it's like a vast wasteland. So you want to avoid all of those unnecessary regions that aren't really
Okay.
that informative. And
Okay.
so that's where attention can help. But what we're finding right now is, like they're not really that good. So maybe thinking about ways to constraining the attention using functional genomics data to focus on specific regions, like masking out the junk DNA regions might be a strategy moving forward.
Wow. There is the architecture that was used, I think GPN,
Yeah,
and as a model, I was as you were presenting, I was thinking about what this architecture is, the GPN, and it was almost like dominant somehow among
yeah.
the CNN based models.
Yeah. So GPN is it's almost like just like if you remove multi-head self-attention and you replace it with a convolution, but with dilated convolutions. So
Wow.
there's just a bunch of convolutional layers with skip connections, and it runs pretty deep.
Oh, wow. That's an engineering revolution.
Yeah, I would say that even for protein language models, these types of convolution-based language models have been shown to be do comparable with transformer-based language models.
Okay. Um, I don't know whether there are questions from other members on the panel, but
Yes, yes, I have a
yes,
question,
Dixon.
please. Thank you, Professor Peter, for the presentation, please. We appreciate you meeting us. My background is in biostatistics, and I've got introduced to genomics. What I like it mostly about is its background and being the cornerstone for precision medicine. So I've got a chance to read your paper, please. And I had this question in my mind. Why do you think one hot encoding outperforms the gLMs in many tasks? Because I've seen it happening in your paper, please.
Wait, could you repeat that question again? I kind of lost the connection just got a little broken up.
Okay, okay, please. Why do you think one hot encoding outperforms the gLMs in many
Yes,
tasks?
yes, and also for me to compliment on that, I think it would be my last question. The one-hit encoding, and this is a technique that personally I despise, that I find like it's preliminary, but it is really performing better in this scenario. And yeah, thank you, Dixon. This is a
Yeah.
good question.
Yeah, so one hot encoding is just some arbitrary representation of nucleotides or biological sequences, right? There's
Yes.
nothing special about it. It's a naive encoding where it's uninformative of the context,
Yeah.
anything related to the sequence. An A here is the same as an A here versus gLM embeddings, which try to encode that A into a different vector considering the context of the sequence. And so it seems natural that the gLM embeddings should in fact be more informative than one-hot encodings. But
Yes.
imagine that instead of encoding meaningful biological like patterns, you don't really capture anything that meaningful. Like there's no
Some
guarantee
information
that the
that is not.
gLMs are capturing anything meaningful in their encodings either. In fact, one could even lose information if if the encoding was less informative, or like ignoring certain regions of the sequence that were actually important. And so this is why we just didn't know. Like we're hopeful with language models, but we just didn't know what they're encoding, and this was just an exploration of what they're encoding. And it seems like they're capturing low-level statistical patterns similar to like Kamer frequencies or something like that. Nothing that high level. And why it underperforms the one hot, well, you know, like every study, everything has to be taken with a grain of salt. These are done on specific data sets with specific models. The models were, while we did a little bit of hyperparameter exploration, just changing, you know, the different numbers of filters in each convolutional layer, we didn't do a full-scale architecture search to see, you know, there might be like an obscure you know, setting where the gLMs can actually be harnessed with, you know, some specific CNN architecture. But I feel like that goes against the principle of pre-trained models, where we don't want to have to do that, because the pre-training should have done most of the heavy lifting, and we should be able to rely on simple models, simple downstream models, and that just simply didn't happen in the study.
Okay, thank you, please.
First of all, I have to say, Prof, this is my best talk ever since we started, and it is the least attended. And incidentally, I don't know. But when we think about that, the encoding of the gene or the DNA, like, do we need to worry about, like in language models, we worry about the position encoding and, you know, all that. Do we need to worry about position encoding for these in terms of the gLM for the different motifs or the individual?
Yeah. Yeah, I think it's really important that we do. Positional encodings are really critical, because like if we focus on just a regulatory element like an enhancer, we know that if there's a protein binding here and there's a motif that's interacting with the protein, and another protein is binding here, if they're sometimes there's some optimal spacing between specific proteins. And as you go further away, the interactions drop off quite dramatically. And so there's position-specific interactions between these things, distance-dependent interactions. And on top of that, that's at the most fine scale of proteins interacting with DNA. Now, these proteins interacting with DNA, the extended regions of like 200 nucleotides to 1,000 nucleotides comprise an enhancer. And these enhancers interact with like promoters, and they also interact with other enhancers. And at that, you know, intermediate scale, the distance between an enhancer and promoter also have strong interactions as well, dependencies as well. Sometimes if you bring an enhancer closer to a promoter, it tends to have a stronger effect, and it drops off as it goes further away from the promoter.
Wow. Prof, lastly, I've not looked at the bottom part of the of the paper down after the references, but my question is about, is the code and data for this work like open, can be looked at?
Yes. We have our code on GitHub, and we have a static version on Zenodo. So there's a link to a Zenodo file or repository where we put all of our data and models as well.
Okay. Zenodo, I'm not aware of Zenodo, honestly.
Oh, it's just it's a repository that allows you to store like 50 gigabytes. And so it's an easy way to like dump your data, share your data and your model weights and stuff like that for free.
And the links are in the paper?
Yes, they are.
Okay. I guess that brings us to the top of our discussion. And I really want to thank Professor Peter and Amber, deliver our thanks to her for this wonderful presentation and the wonderful work. I'll stop the recording. And we really thank you. I know professors are busy people, but you dedicated time and you really decided to talk to us. Thank you very much, Prof. And I would say, like, thank you. I can't thank you enough.
Yeah. I mean, thank you for the invitation. This was, you know, this is actually the first time I gave a talk on this work. I typically give talks about other things, and so this allowed me to